Good morning, everybody. Wow. Hopefully you can hear me. Welcome to the 38th Massie lecture, and it's my distinct pleasure to introduce Chuck Murry, who is a longtime friend who I've known for 20 years. Chuck is a great selection for this lecture, similar to Edward Massie, who pioneered the electrocardiogram and was a staple here at Washington University. Chuck has also done a lot in the field of stem cell and regeneration, and specifically how it affects the electrical activation of the heart as well. So they have that in common, and he's been a staple at the University of Washington for many, many years, decades. Like a barnacle. They haven't removed the barnacle, right? So lucky for them. So Chuck first started out in North Dakota. He's from North Dakota. He was then at Duke, did his pathology training at the University of Washington, where he became faculty after that and has remained there and rose through the ranks and became full professor in 2004. He's also the director of the Cardiovascular Institute and the Stem Cell Institute at the University of Washington, and he's a leader in the field of stem cell and regeneration. So Chuck, as I know very well, has been very brave throughout this whole journey with stem cells. He's not afraid to challenge dogmas, and when the next big experiment needs to happen, Chuck is there to do it. I remember that a lot of experiments were happening in small animal models, and Chuck did a lot to push forward in a non-human primate model, the stem cell field. And this has led to a lot of great discoveries that are very translational and relevant that I'm sure you'll hear about today. Chuck is the recipient of many awards, which you can read here in the pamphlet. And one thing that you won't read in the pamphlet is that he really is a mentor to so many people, not just in his own lab, but also across the country. And I can tell you personally that I've known Chuck for 20 years, and he really cares about the younger generation in the field. And he doesn't just care, he actually is actionable with that care. And he's the kind of person who, when you have that wild idea and you need somebody that you trust to run it by, that would be Chuck. So I really look forward to hearing about his latest and greatest. And with that, welcome.
Thank you, Stacy, for that way too generous of an introduction. I think I'm doubly mic'd. I'm just going to bend that one over just a little bit. And it's come full circle because now I call Stacy with my crazy ideas as well. So it's great to it's at least mutually beneficial. Stacy, thanks so much. Delighted to be here at Washington University in St. Louis, the place that the University of Washington is often confused with. I don't know if you all get this, but we get this all the time. So how long have you been in St. Louis, Chuck? That sort of thing. Anyway, I'm delighted to be here. It's been about a decade since I've had a chance to come by, and really a privilege to be named as this year's Massie lecturer. So thank you very much for that honor. What I'd like to do in the next 45 minutes or so is really give you some of the scientific foundations for the field of heart regeneration. I'll start first with some of the natural biology of the mammalian heart, what we know about cell turnover, what we don't know, that sort of thing. And then I'll move more into focus on my own work in stem cell-based heart regeneration, some of the challenges and the recent progress that we have made. These are my disclosures. And so I'm going to start by telling you that the times are changing. That the world of stem cell and gene therapy is a thing now. We actually have FDA approved, and I think this is going to become the third leg of the practice of medicine. The first, of course, being small molecules, things chemists could make. The second being large molecules, things where we harness cells as factories to make protein therapeutics. And then the third is going to be cells and genes. And this is progressing really towards living therapeutics. So a couple of predictions here. The first is that this is going to be revolutionary. This is going to change the way we practice medicine. I earnestly believe that. The second thing is this is going to be a slow revolution. Our imaginations took us way ahead of what we were able to reduce to practice with our hands. This is like welcome to humanity. This kind of stuff always happens. But I think that this is worth continuing, because I think we will be able to reduce these most complex medicines ever, and change the way the heart heals, for example. This is an introductory slide, a Cole to Newcastle slide. Basically, I think you all know that the heart is essentially the least regenerative organ in the human body. There's so little regeneration that basically with an acute event like a myocardial infarction, there's essentially no cardiomyocyte repopulation. The heart heals by scarring, and we go into this downward spiral of decompensation that too often ends in heart failure. And you all know that once you're diagnosed with heart failure, it's worse than most diagnoses of cancer, and you have on average about 5 years to live. And so in its simplest form, our idea is that this is a classic disease of cellular deficiency. We can now make essentially inexhaustible amounts of human cardiomyocytes in the laboratory. We should be able to remuscularize the heart. That's been my notion for more than 2 decades. And so, as I said, it's been a little more complicated than I ever would have imagined. So let's talk first about how the mammalian heart behaves. The first thing is that we're born with regenerative ability in our hearts. The heart isn't completely baked, if you will, when we pop out. And we learned this best probably from this old paper now from Enzo Perello, when he was working with Hesham Sadek at UT Southwestern. But what they did was to amputate the tip of the heart, the apex of day 1 neonatal mice. And if we look in here, zoom in, you get a clot that forms and you get hemostasis that way. But gradually, cardiomyocytes regenerate and repopulate. And instead of scarring, the scar almost sloughs off like a scab at the apex, and you get complete regeneration. If you do this same experiment at day 3, no regeneration. So there's this window that we seem to be born with. More recently, work out of Jay Zhang's laboratory in Birmingham, Alabama, has shown that neonatal pigs have the same regenerative properties. This is a typical infarct. These are, you can't show the whole heart as elegantly as you can a mouse heart, perhaps, but this is a trichrome stain where collagen is red. Typical infarct here in a pig that was 14 days postnatal, and you can see the thin wall and the scar tissue. Whereas if this animal was infarcted at 1 day postnatally, you can see there's been regrowth of new myocardium. And conversely, here we're looking at scar deposition. You can see the longer one goes postnatally with the coronary occlusion in the pig, the greater degree of replacement fibrosis that happens. So this has become a really useful model to study endogenous mechanisms of regeneration. And then the question is, why does this get shut down? I don't have the answer today. If there's audience members who have ideas, I am all ears, because I'd totally love to know. So what about humans? Well, the first thing is that when we're born, our cardiomyocytes are small and they're diploid, so they have a 2N nucleus. And then there's tremendous physiological hypertrophy that happens as the heart grows postnatally in human beings. Our cardiomyocytes get 20 to 40 times larger. Apparently, you can't grow that much with just a single genome. So throughout the mammalian kingdom, if it's not a kingdom, but whatever, throughout mammalia, we have polyploidy that serves, so the cells have extra copies of the genome, and presumably in order to do that much protein synthesis. This is accomplished in different ways. In humans, there is DNA synthesis without nuclear division, and so we have polyploid nuclei. And in rodents, we have DNA synthesis, nuclear division without cell division, and so we have multiple nuclei in the cell. But the bottom line is about 75% of human cardiomyocytes in the adult are mononuclear and polyploid. Now, what happens with growth and disease? This is some old work from a pathologist named Adler from the University of Freiburg in Germany. And what we know is that as heart rate increases, there's a direct linear increase in the number of non-cardiomyocytes. And so think fibroblasts, vascular cells, that sort of thing. So regardless of what the heart rate is, and this is, I put the arrow here to show what the normal, this is left ventricular that's been trimmed down just to the meat, if you will, of the chamber. And so normal left ventricular weight's maybe 175 grams. And so whether it's through physiological growth or pathological hypertrophy, we can easily make more stromal vascular cells. It's more complicated if we look at cardiomyocytes. In here, Dr. Adler had to count nuclei rather than count cells. But what you can see is early postnatal life, there is a steep increase in cardiomyocyte nuclear number with physiological growth. And then we hit this plateau phase up through where physiological hypertrophy ends and pathological hypertrophy begins. And so that's increase in cardiomyocyte mass to accommodate organ growth. And then as we get into the phase of pathological hypertrophy, and this is the part I found most surprising, there is a little bit of increase in cardiomyocyte nuclear number. And I've played around with the math in my head, and there is no way I can make this happen without cell division, or at least new cardiomyocyte generation. So there's a little bit of this. It looks like it's extremely slow, but it does happen. So there's a difference between being no growth and slow growth, and slow growth pathways can be discovered and exploited. So there's been this evolutionary loss of the ability of the heart to regenerate. If we look at zebrafish, newts, and axolotls, they're masters at regeneration. They can regenerate their spinal cord, they can regenerate their eyes, they can regenerate their hearts. And then in mammalia, this has been lost. There's this neonatal window, and then afterwards, we don't have it. And there's a lot of interesting work going on. Current ideas are focusing on metabolism and thyroid hormone signaling and that sort of thing. But the truth is we don't really know why we lose this, or why it's apparently advantageous to lose the ability to regrow this very valuable organ. But we would like to undo this and focus on various aspects. Today is going to really be about remuscularization, but we also have to figure in the electromechanical function. We have to do, what about all the fibrosis that goes on in the heart, the immune cells, both innate and adaptive, and then the vascular system as well. So it behooves one to think holistically, but then it's hard to reduce everything to practice all at once. So the basic pathways are first remuscularization. How is it that we're actually going to replace the lost cardiomyocytes? And for this, there are three ways that I can envision. The first is through cell transplantation, either transplant monodispersed cells or engineer the cells into a tissue ex vivo and then transplant this on as, say, a sheet onto the epicardium, which is being done in a number of locations. The second pathway is to stimulate endogenous regeneration of the cardiomyocytes, activate the cell cycle in some way and grow your own, if you will. And then the third would be to reprogram cells that are not cardiomyocytes today to become cardiomyocytes. And we're learning more about how to do this, turn a fibroblast into a cardiomyocyte, for example. And yes, the stromal vascular cells of the heart are important. As I showed you already, they proliferate quite well. They're very important in terms of crosstalk for tissue homeostasis. No tissue is a monoculture, cartilage perhaps, but essentially no, certainly the heart is not. And it's like a symphony, this conversation that's constantly going on. And the mantra we have in my laboratory is that we're going to have to revascularize if we want to remuscularize. That said, today, this is going to be a myocyte-centric talk. In the vascular work and in the connective tissue and so forth, we'll have to wait for another time. So I'd like to just share a few things that have been going on outside of my laboratory in this field of regeneration that's not using cellular transplantation. And there's been a breakthrough really in understanding what it is that makes the heart stop hyperplastic growth. And that's this pathway called Hippo. It was originally discovered in Drosophila, and it's the basis for what cell biologists have long known as contact inhibition. When two cells come into contact, this tends to slow their growth. And so this is work out of Jim Martin's lab that I'll be quoting initially. And the idea is, of course, if you could get proliferation, you could take a scar and regenerate the myocardium. The Hippo pathway is really, the terminal part is just a short kinase cascade where A phosphorylates B, phosphorylates C, and that results in inactivation of a transcription factor called YAP. So YAP goes out of the nucleus, and YAP is a very powerful pro-growth signal. So when YAP is pulled out of the nucleus, this seems to be like the linchpin for making the cardiomyocyte stop dividing. So Jim Martin and others have been targeting the distal elements of this pathway and taking it up the food chain, if you will. And there, I'll just show you something from a recent paper that they had in Science Translational Medicine, where they did this in pigs. And what they used an adeno-associated virus to add a dominant negative to this Salvador, with a cofactor here for this kinase. And this blocks this kinase pathway up here, so YAP can then go into the nucleus. They put this into the pigs, and they found that they treated the animals at 14 days post myocardial infarction, and you can see the low and the high dose animals showed recovery of their ejection fractions, whereas the control group showed progressive dysfunction, let's say. And what they found when they looked structurally, and this is, I could have shown you the histopathology, but you can actually discern it with your naked eye. There's a transmural scar in the apex from the LAD occlusion in this heart. And in this one, you see these little bridging strands of new myocardium that have been grown back across. And they did all the controls to make sure this was new, not pre-existing myocardium and that sort of thing. So really powerful pathway. But it's a tricky pathway. It's kind of like playing with fire right now, because Mauro Giacca's group tackled this just a slightly different way by using a microRNA that antagonizes multiple branches in this pathway. And they also found regenerative growth. They found improved ejection fraction. And then they found this, which is small blue cell tumors that were growing in the hearts. And in pathology, this is just, it means, undifferentiated malignant neoplasm, not otherwise specified. And you can see, if you look at the survival curves, when these started occurring, the survival crashed in these animals. So I think, playing with fire is essentially human, right? This is what we do, but we're going to have to be smart about this before we start in people, because of course the last thing you want to do in a patient who's had a recent MI is to give them a cardiac tumor. But I think it's a really interesting pathway. The other approach, briefly, that I'll go through is direct reprogramming. The idea here is that you start with a fibroblast that's been activated in the heart by the myocardial infarction, and you can actually change its fate. And this was work originally that came out of Deepak Srivastava's laboratory. And if you overexpress a cocktail of transcription factors, you are able, that are characteristic of the cardiomyocyte, you can change the cell's fate. And then they make these what they call induced cardiomyocytes. And they're shown here are various ways to transfer the genes into the cells. And if one does something like this, is from Masaki Aieda's work, you can see a field of myocardium where the fibroblasts have been labeled red with genetic lineage tracing. And we see in the center here a cardiomyocyte that has been induced by this pathway. So the fact that it's red says that it's got the lineage trace to tell you it used to be a fibroblast. So really quite interesting. And if you do this genetically in a mouse and turn this these transcription factors on in fibroblast post-infarction, you can see that ejection fraction starts to creep up, and you can see separation with the control group. The rub with this is that it's very inefficient at the moment. The biological proof of concept is there, but we're going to have to get a log or two of increased efficiency before I think this will have real therapeutic relevance. So we'll spend the rest of the time talking about transplantation of cardiomyocytes, which is what my laboratory does. The concept is deceptively simple. Let's manufacture cells, let's deliver them in a variety of ways, and then the heart does the rest. The heart is the bioreactor, and it lets the cells incubate. And this is beautiful maturing myocardium human myocardium that's been in the heart of a non-human primate for 3 months. Delivery is the best ways to do this is either with direct surgical injection or with a catheter that's got like a little stiletto type needle that allows you to do transendocardial delivery. Manufacturing has evolved a lot over the last 10 years or so. This is when we first did a non-human primate experiment. This is what we did to grow our cells. It was just a brute force effort with a lot of great people who were doing this work. But we had essentially a tennis court surface area just to grow the cells in 2 dimensions. And this is conditioned media. You don't even want to know how anyway, it just suffice it to say it was like moving heaven and earth to try to make this happen. Today, it's a little different. This is from our clean room and our good manufacturing practices laboratory at the University of Washington. And these are a couple of our staff. You can't tell who is who because they're in moon suits at this point. But we can grow these cells under GMP conditions, and really they're a pharmaceutical grade. And we struggled for a long time to learn the secrets of how do you make cardiomyocytes. And then in the end, it became quite simple. It's all about the Wnt pathway. And so initially you start with a pluripotent stem cell, you activate Wnt to make mesoderm, and you repress Wnt, and it activates the cardiac pathway by default from there on. You just let it go, and you get beating cells in 7 to 10 days. And we typically harvest our cells 3 weeks or so after the induction of the Wnt pathway. This is a stirred tank bioreactor. What you're seeing here is a 3-liter paddle wheel type. And if we stop, you can see like the world's most expensive snow globe. This is about $80,000 worth of reagents that went into this. And I'll speed this up so you can watch this settle. But it has about 8 billion cardiomyocytes in it. So that's a couple of left ventricles right there growing inside. Now, playing the part of the snow in the snow globe are these little cardiac organoids. And if you look at them and just watch for a moment, what you'll see is each of these little spheroids is a beating mass of cardiac muscle that has about 1,000 to 2,000 cardiomyocytes per little cardio ball there. And if we zoom in higher, it makes it easier to see that they're individually contracting. And whenever I get depressed, I like to go watch my cells beat, because it's just like, you know, if nature has done this for us, we should be able to figure out what to do, you know. We can engineer these in a variety of ways because they start with stem cells. Here we've popped in a transgene called GCAMP to allow us to follow the calcium transients inside the cells. And so we have what we call flashing beaters. And the idea is here that we can do this so that we can transplant the cells and use it like a little space probe signal or something that comes back and says, I'm alive, I'm well, I'm beating, that sort of thing. So we did that, and we transplanted these cardiomyocytes into the heart of a non-human primate. And what you're looking at here is a black and white image of the probably apical 25% of a monkey's left ventricle. And if I start the video, it may take a minute for you to kind of get to see it, but there is cyclic fluorescence, probably seen best here. And then you start seeing it's sort of a whole field over here and here and here. And what that is the calcium transients of the human myocardium. So that says that months after we've transplanted these cells in, they're alive, they're beating, and at least the graft cells all seem to be beating in synchrony. And so if we take this out and we do image analysis on it, you can see these synchronized calcium transients, and that's what your eye already told you. But what your eye can't tell you is what's going on with the rest of the monkey's heart. And here's the electrocardiogram from that. And you can see there's perfect one-to-one correspondence between the electrocardiogram driven by the sinoatrial node and the human cells that are flashing in sync. So it says that these have done exactly what we'd hope, which is to remuscularize the wall of the heart, integrate and beat in synchrony with the sinoatrial node driving it. It doesn't always work that way. I'll come back to that. So histologically, this is a non-human primate heart 3 months after we've transplanted human cardiomyocytes in it. We infarcted this animal, waited 2 weeks, and then came back surgically with needle and syringe and delivered 750 million human cardiomyocytes. And in blue is collagen. It's a little hard to see the collagen because we've remuscularized this reasonably well. So there's quite a bit of new human myocardium here. And this is simply a mass in, mass out relationship. We put in 750 million cells, and we got, gram amounts of new myocardium to form. You might imagine this could improve the squeeze of the heart. And indeed, if we look at the ejection fraction by MRI, a non-human primate is, say, 65% on average. The reds are the control animals that receive vehicle, the animals that receive the human cardiomyocytes. These are all immunosuppressed, by the way, triple drug immunosuppression, because this is xenotransplantation. So there's nothing immune privileged here. In any case, you see that post-infarction, we lose about 25 points of ejection fraction. And then in the controls that receive a vehicle injection, there's no improvement even out to 3 months. But we get about 10 points of improvement at 4 weeks. And in a couple of animals that we let go further out, we get another 10 to 12 points. So back up into the normal range. And I've been at this game since 1984. I started in heart research, and I have never seen anything that could do this to an infarcted heart. So this is the thing that gets me out of bed in the morning. Because human cardiomyocytes can do this for a non-human primate, they should be able to do this for a human. This is a thing that keeps me up at night. And there's a toxicity associated with this, a period of electrical instability that we've called engraftment arrhythmias. And they manifest we're going along here in lovely sinus rhythm, and then you see this wild, ugly, complex, polymorphic, wide complex of ventricular tachycardia. And this has been, we never saw this in mice, rats, or guinea pigs, probably because their hearts were too fast. It was only when we went into the non-human primate that we found this, because their heart rates are about 120 beats a minute. And this is typically, what happens is you have the vertical line here is when transplantation takes place. The animals go into a period of electrical instability, but then they come back out of it. It takes, on average, a month or so to come out. Same kind of thing has been seen when monkey cardiomyocytes are transplanted into the monkey heart. So it doesn't seem to be a species dependency here. And Mike Laflamme's group showed this also, that when human cardiomyocytes are transplanted into the pig. The monkey heart seems to tolerate, the monkey seems to tolerate having this kind of tachycardia, even though the rates are up at, say, 250 beats a minute. It's a small heart. I think it doesn't, emptying and filling, the dynamics are different in a small heart versus a big one. Pigs don't like this. And the couple of arrows that I put in here indicate 2 animals that had sudden cardiac death as a result of this. So pigs are more sensitive. We don't know how a person would be, but this is not to be taken lightly. And so we started to explore this. We wanted to know, what's the mechanism of this? And again, I don't think I need to remind this crowd that there's 2 general mechanisms for arrhythmias. One is impulse generation, and the second is abnormal conduction that leads to what's called a reentrant pathway. And so that was, which of these things were we dealing with? Because, frankly, my money was on reentry. But when we map this out electrically, this is a low resolution map of electrical activation. The aortic valve cusps are shown here by these balls. So the base of the heart is here, the apex is down to the right. Typically, the heart should activate at the apex, and depolarization should progress towards the base where there's outflow, right? We're activating here in the anterior septum. We couldn't see anything that looked like a rotor, low res map, but the monkey heart's like the size of a plum, and so it's a smallish heart to be doing these kinds of studies in. And we've done I'll stop this, it's distracting. We've done a variety of studies in this to try to figure out, is this reentry or impulse generation? And so mapping, as I showed you, favors automaticity. We tried to pace our way out of this. We could capture it, but as soon as we turn the pacemaker off, it would go back to its original arrhythmic rate. That favors automaticity. We tried DC cardioversion, so we tried to shock our way out of this. There's a brief pause and the tachycardia resumes. That favors automaticity as well. And then when the animal was in sinus rhythm, because they go in and out, we brought it into the laboratory and tried to do program stimulation to induce it. We could never induce it. And typically with reentry, you can do that. So all of this favored automaticity. And we had we had to make a call here. And this is clinical grade evidence, I would say. We usually like to have something like genetics to be really sure. But we had to make a move at the fork in the road, and we decided to pursue automaticity and not focus on reentry as a mechanism. We also pivoted to pig at this point. It was impractical to try to do these studies in the non-human primate, and anything you, for obvious reasons, anything you could do in a pig, you should probably not do in a monkey. So we developed technology that everybody here will understand well. For percutaneous access, here's a coronary angiogram. This is the left anterior descending artery of a pig. Here's the left circumflex coronary. And then we used a balloon catheter to inflate, and you can see it blocks flow in the distal LAD past the first diagonal. And when I say we, I didn't do any of this. Like anything I talk about today is not me, right? So when I say I think we, when I say we, think they, right? Just to translate, so Ken Nakamura is a really good interventional cardiologist who's been working with us for about 5 years now. He's terrific. And Lauren Neidig is a large animal veterinary surgeon. And so it's Lauren and Kenta who do all this stuff. Anyway, so we have a pig model. And then we also have developed MRI-based imaging for both the non-human primate and for the pig. And this is work done by Anna Naumova. And you can see here's a delayed gadolinium enhancement of an aneuryseptal infarct in the pig in both short axis and long axis views. Okay, so then we typically wait a couple of weeks, let the infarct go to the granulation tissue phase of repair. We come in, here we delivered cells surgically. Here's the LAD of the pig heart, and you can, if you squint, you can see these little puckers in the infarct zone that have purse string sutures that we cinch, where we cinch up our injection sites to keep the cells in place. We also can do catheter-based delivery. This is the Noga MyStar system from Biosense Webster. Sadly, has been discontinued, and so we've had to switch to almost exclusively surgical delivery now. And we get these nice grafts of human myocardium in the pig heart. We can here identified with human-specific troponin antibody, and this is nice maturing human myocardium like we saw in the immunosuppressed monkey. These are heavily immunosuppressed pigs as well. And so the first thing we wanted to do was to say, is there anything in the pharmacopoeia of antiarrhythmic drugs that will make this go away? And of course, this is totally an iatrogenic problem. This is not an arrhythmia that has existed in nature before. And so we didn't know really whether anything that the FDA had approved would work. But we brought animals in while they were in engraftment arrhythmia, and we gave them sort of acute boluses in the laboratory to see if we could get anything. And the biggest hit we got was with amiodarone. Amiodarone is double-edged. It's so nonspecific. It gets so many different ion channels that it's really dirty from like a mechanism of action standpoint, but it's kind of a good for what ails you tonic for arrhythmias as well. So here's 3 different animals where you could see we actually broke the VT and we got partial conversion to sinus rhythm. So that was cool. And the other thing that we found was Ivabradine. Ivabradine, you're probably aware, targets the HCN4 channel that carries the funny current that is often considered to be like the pacemaking current. And that didn't break the arrhythmia, but it slowed it way down. And it's not so much that the animals are in have this ectopic pacemaking that's the problem. It's really the rate. I think you start to get into this poor coronary perfusion because the diastolic interval is diminished, that sort of thing. So I think that's really what gets us in trouble. So these became the 2 drugs we wanted to test in a more detailed approach. So we put these animals on, we set up proper control and treated groups and studied the effects of amiodarone or ivabradine combined treatment. And the first thing we found is that we got better survival. So this was wicked. When we lost 2/3 of the pigs, and most of them we had pre-specified euthanasia criteria, and we would, but still, this was a deadly arrhythmia that we had set up. And you can see that if we have all animals that we included, the so-called intention to treat design, we get borderline statistical significance. These 2 animals died of pneumocystis and porcine cytomegalovirus. So I asterisk that. And if you then if they say, well, what about cardiovascular survival? We had 100% cardiovascular survival compared to 40% say cardiovascular survival. So that was a big deal. What does it look like in terms of like the burden of the arrhythmia and the effects on heart rate? So what you can see, the infarct is the first dotted line, the cell transplantation is the second dotted line, and the heart rate on average shoots up. The fact that this is group average belies how high some of these individual animals' rates get. As I said, 250 or so is common. And then it starts to come back down as the grafts mature. And the animals that got this combined treatment, they get a little tachycardic, but it's nothing like what happens in the control group. And if we look at the burden of the arrhythmia, and by that I mean the fraction of the day that the animal spends in arrhythmia, you can see that in the control group, there's maybe 75% of the day at peak, and we have about 50% of the day at peak. So the area under the curve here is reduced, but the main thing I think really is rate control. So that was encouraging, but we wanted to I wanted to know what the mechanism was behind this, and so we started this thing that came to be called Project Medusa. And Medusa is an acronym that stands for Modifying Electrophysiological DNA to Understand and Suppress Arrhythmias. And it's been led by this great team of cell biologists. Sylvia Marchiano is the postdoc. And the idea was, let's go after automaticity. And moreover, we wanted to, the goal was really to make cardiomyocytes that lacked automaticity but remained electrically excitable. And so the workflow was basically to identify all the world's ion channels, take the ones that, borrow from the literature for what we know about cardiac electrophysiology. We did an RNA sequencing experiment to help us understand what happens during maturation, come up with a prioritized list, and use gene editing to get after this. And so we made gene-edited cells. We would test them in vitro, and then we would test in vivo. So this is where it turns into the world's slowest CRISPR-based screen, because the only thing we could do to test in vivo was in a large animal. So we had to screen in the pig. So we would have to make 150 million cells per animal and then see if they got engraftment arrhythmia. And the answer was almost inevitably yes. So here's the pedigree of all the different cells that we made. We start out with wild type cells. The swirly-eyed pig means you get engraftment arrhythmia. Here is all the ion channel genes that we knocked out in singlicate. The next tier down means we did things in doubles. The next tier down means we did things in triples, and finally in quadruples. And obviously things didn't work with just one, or we wouldn't have gone to all of this combinatorial stuff. So let me just, and I won't run you through all of this because it took a long time, but I'll start just with HCN4, which carries the funny current. So we're essentially hacking the genome at the level of what people have called the voltage clock. This is the HCN4 channel, which carries the current that's a diastolic depolarization current. And we knocked this out with CRISPR. We validated by patch clamping. This is actually work from Bjorn Nolman's lab at Vanderbilt, validated for us that we actually had lost this current. And we transplanted this, and it failed. But let me just run you through this so you can understand what the studies look like. In black is the control group, and that received wild type cells. And you can see here we're looking at the fraction of the day that the animal's in arrhythmia. And you can see this goes up, and then it starts to come down as the days go on. But we're losing animals along the way. Every red dot is an animal that we've lost. So in the end here, we've only got two. One resolves its engraftment arrhythmia, and the other remains in persistent engraftment arrhythmia. And we really have no significant change in that when we knocked out HCN4. Skipping the next 2 years of pain, we now have swirly-eyed pigs all around this cellular pedigree, where we've tested things in vivo and nothing is working. And Sylvia's like, Chuck, you told me this was going to be a great, this is a career-making project for me, and am I ever going to get a job? And so if you look down here, we had made a single, double, triple edit, and we wanted to go ahead and do a quadruple edit. That was the thing. Let's do this one more college try. And so what I'm going to show you is data from where we had hacked the voltage clock, basically. We'd taken out two depolarizing channels here, HCN4, and then a T-type calcium channel. And we've overexpressed a repolarizing, hyperpolarizing channel called KCNJ2. And then we also took out the big kahuna, which is a sodium calcium exchanger as well. I was hesitant to do this, but in the end, we just wanted to understand mechanism. So we took out the sodium calcium exchanger as well, because this is also a depolarizing current. And we came up with cells that have these 4 edits that I just mentioned. And from henceforth, I'll just call these the Medusa cells. And it was very interesting. Here they are in a multi-electrode array. They're completely quiescent. These guys are very chill. They're cardiomyocytes, but they don't beat in monolayer. If we do patch clamping on them, these are wild type cells that we're pacing at 1 Hertz. You can pace them and get action potentials at 1 Hertz. They follow up to 3 Hertz, which is as hard as we push them. But the action potential has different characteristics. Most notably, it's missing this diastolic depolarization current. The diastolic phase is flat here, like an adult ventricular myocyte. So we were trying to hack the action potential to make it look like an adult cell. And the other thing is that its resting membrane potential is more negative. So you can see here in a couple of different ways to look at the resting membrane potential. It is more negative. So we had our desired outcome. We got cells that were excitable and yet not automatic. And we transplanted those in and we saw dramatically different results. Here's the same control curve I showed you before, but 3 separate animals curves are shown here along the bottom. There's very little arrhythmic activity. And if we take the worst of these, this guy here that's in sort of maroon, and unpack it, what you can see is that it's really, there's no sustained VTAC, and it's just PVCs and a few short salvos of non-sustained VT. And so this stuff here that comprises most of that is really just PVCs. Overall, if we get 90 to 95% reduction in engraftment arrhythmia. So to summarize this phase, what we did was we transplanted in initially the wild type cells that have these 3 depolarizing channels, and we knocked them all out, and we put in this one hyperpolarizing channel that's characteristic of an adult cardiomyocyte, and we made these cells that were quiescent yet excitable. And when we transplanted them in, what I haven't had time to show you is they do beat, and they beat in synchrony with the rest of the heart, but they don't cause this terrible engraftment arrhythmia. So thanks to the advances with genome editing that have come along, we've been able to figure out at least the mechanism of what's causing this arrhythmia. We know the culprit ion channels, and this might be a useful way to make a cell that doesn't cause it. I know time's getting short, and so I want to show you just a quick peek into the future, which is we'd like to switch from treating acute infarcts to treating a chronically failing heart. And this is something cardiologists always ask me. I'd say, hey, look, we've figured out how to keep the heart from going into failure. And they're like, oh, prevention? Can't you make something that works when people have really advanced failure? Those are really the patients we don't have options for. Well, that's harder. So we've been at this for a long time. And the first thing is that we've tried it hard to make wild type cardiomyocytes work. And this is an experiment done in rats with human cardiomyocytes where we have absolutely no impact on fractional shortening once failure is established. Same thing in guinea pigs. We have absolutely no impact. We get engraftment, the cells will beat in sync, but perversely, they do not improve systolic function. So what's going on here? This remains enigmatic to me. Why does this work in the acute phase? It doesn't work in the chronic phase. It's clearly a more complex substrate that we're dealing with. Yes, it's a problem of myocyte deficiency, but it's also there's a number of other things that are going on. Whatever paracrine things that are going on with the inflammation and so forth in the acute phase of injury, that window's probably closed. The thing I really want to focus on today is the fact that failing hearts have failing myocytes, and the rest of the myocytes are badly dysfunctional. And also scarring is complete, and the ventricle has changed its shape as well. But the fact, inspired by LVADs and the things that they can do for reversing heart failure in some patients, I really wanted to focus on mechanics. And so if we could engineer our grafts in such a way that they would not only remuscularize, but also restore contractility to the rest of the heart, this might be a good way to improve ventricular function. So this is work that I've done, been collaborating for 15 years or so with Mike Grenier, who's a bioengineer at the University of Washington, and a postdoc named Ketki Montre, who really led all of this work. And it all comes down to a simple molecule that everybody's heard of, which is deoxy-ATP, one of the 4 building blocks of DNA. Everybody knows plain old ATP is the substrate for myosin. It's the energy for contraction. It turns out deoxy-ATP is like a super substrate. Not only can it provide the energy for contraction, but you get stronger contraction and you get faster contraction. This is because it's an allosteric activator of myosin. And I won't get into the biophysics of how this works, but in every species where we've tested this, this will improve contractility. Importantly, even in failing human myocardium, where we've taken strips out ex vivo and bathed in deoxy-ATP. So, kind of interesting. It's made by an enzyme called ribonucleotide reductase. This is a rat cardiomyocyte that's being stimulated to contract at 1 Hertz, and it's been hit with a control virus. And just watch it for a second so you can get an idea for what normal shortening is. And then here's one that we've hit with ribonucleotide reductase. I think you can appreciate, like, there's better shortening in the guy on the right. And if you can quantify this here, that with each twitch, if you will, you get maybe double the shortening. So that's for the cell that gets the enzyme. But the cool thing here is that deoxy-ATP is a small molecule, and the heart is the most gap junction network organ in the body. And so deoxy-ATP can diffuse out from one cell into the next. You can get secondary, tertiary, quaternary transfer. And it gradually, we found in culture that it gradually spreads. So shown schematically here, we did this by putting transduced cells in culture with non-transduced cells and looking at pairs. And if it's just a GFP virus, both cells have the same contractility. But if it's ribonucleotide reductase, both cells have augmented contractility. So here was the idea. We could take the chronically failing heart, an ischemic cardiomyopathic heart, this would be an athymic rats, and transplant in the deoxy-ATP producing cells that we'd engineered in the laboratory, and that this would seep out into the rest of the heart through diffusion and gradually improve contractility in a setting of chronic failure. So we did the engineering of our stem cells with, again, CRISPR-Cas9 based editing. And this is for the first thing we asked was, do we actually get spread of deoxy-ATP? Here is the apex where the graft is much higher deoxy-ATP than in controls. But in the next layer up, which we call the border zone, and in the remote, we have increasing amounts of deoxy-ATP. And this is really potent. You just trace amounts, since this is an allosteric activation, are sufficient. So here's what happens when we did this in a therapeutic context. We infarcted the animals, waited a month, put the cells in, and then waited 3 months and studied them along the way. So let me call your attention first to the 2 control lines, the animals that received no cells or the animals that received wild-type cardiomyocytes, virtually indistinguishable. As I showed you previously, there is no impact of transplanting in these wild-type cardiomyocytes. But you can see here, when we transplant in the deoxy-ATP donor cells, we get an inflection, and function starts to improve. Another way to look at this is use each animal as its own control. Say, ask how much its fractional shortening has changed, and you can see things get worse and worse in the 2 control groups, and they're creeping up better and better in the animals that receive deoxy-ATP donor cardiomyocytes. Now, patients don't care so much about what their fractional shortening is. They do care about their exercise tolerance, their ability to carry out their daily living activities and so forth. So as a surrogate for that, we put running wheels in, put the animals in individual cages and gave them wheels to run in. We go through 1 light-dark cycle to let the animals adapt to it and make measurements in the second nighttime. And they're, of course, nocturnal, so they run at night. And what we saw is that we get about 3 times the amount of voluntary running in the animals that receive the deoxy-ATP donors. And so that seemed really quite interesting to us. Patients also care about whether they live. And we weren't really expecting that we would see a difference in survival, but we lost about 40% of the animals in the months post infarction in the untreated control group, and we had 100% survival in the group with deoxy-ATP donor cardiomyocytes. So let me it's time for me to wind up. So let me do that. What I've shown you in the second phase is there's this new class of inotrope that we're really keen to exploit. It's a natural molecule, deoxy-ATP. And we know really well what the mechanism of action is for this. And one of the things that is important, yes, it's an allosteric myosin activator, but it's calcium independent. So unlike cyclic AMP generators and things like that. This will not, we hope this will not have the arrhythmic complications that we've seen in patients that have been treated with inotropes. And it augments contraction, and it doesn't interfere with relaxation. That's also very important. We can increase whole heart levels by creating a relatively modest sized graft of these deoxy ATP donors that diffuses out through gap junctions. And we get this boost in contractile function, and this improves exercise performance and things like that. So this is something we're looking on at taking forward. We'd like to take this into the clinic, like pivot from thinking of acute infarction to really patients with chronic failure who have relatively few options. So to come back to the prediction that I made at the beginning of this lecture about this being something that has the potential to change the way we practice, I don't know of anything in medicine. I don't know of anything in biology, and I don't know of any reasons in fundamental chemical thermodynamics why this should not work. I've been trying to think very critically about this for a long time, and it's my notion that one can be rationally optimistic about taking this forward and making impacts on patients. With that, I'd like to thank the people who really did the work. I tried to name people individually along the way, so I won't do that here, but it's a great group, and I'm really fortunate to work with them. And colleagues at the University of Washington, Mike Grenier, Rob McClellan, our head of cardiology, Nate Snadecki, really close collaborators who make things happen. And then I'd like to acknowledge my funding sources. This took a lot of money, and people have been very generous towards our program, and I'm very grateful. And with that, if there's any time left, I'd be happy to take a few questions. Thank you so much for your attention.
Great talk. Thank you so much. We do have time for a few questions. Doug?
So before we mark this off, I want to go back to the producer project to try to get the postdoc a job. So as you know, with the iPS cells, there is a mixture of mature and different cell types. I'm sure you've thought of it. Some of the cells in your iPS cells are probably pacemaker cells. And is there a way of purifying the iPS cells so you don't have to do all this complex genetic engineering? Just sort of pick out the very big, if you will, the ones that have more of an adult ventricular myocyte phenotype, and then put them in, because the genetic engineering from an FDA perspective is going to create all kinds of issues.
Yeah, thank you. For people on Zoom, what Doug said is there's heterogeneity within these cell preps, that there are ventricular atrial pacemaker cell types and things like that, as well as like temporal heterogeneity, things that are immature and mature. No doubt, despite our best efforts, I mean, the only pure population is like a single cell in the end. And so is there some way that we could select this out? So James Chong from Australia, who's actually a former trainee of mine, just has a paper that's coming out where they did just that. They showed that the extent of arrhythmia correlates with the percentage of atrial or pacemaker-like cells that you have. The more of them you have, the worse it is. I think everybody is a pacemaker at this stage, but the more definitively pacemaking you are. And so it's a great approach. There should be ways that we could do selection. I will say that these cells were selected with lactate, which is one approach. But it's a great idea, Doug. Yeah, simplify it. He just to dig in, he added one more complexity. Thank you for that.
Great talk, thanks. So I was curious about the myocyte non-myocyte calculation in terms of changes sort of above normal hypertrophic growth. So it seemed like non-myocytes do their massive increasing, and also myocytes. You mentioned that based on population, there's kind of no way to figure it out without myocyte division playing a role. So what I'm wondering is how you take into account heterogeneity in the size of myocytes. If myocytes are hypertrophying as a part of the growth, what we normally expect, then that's one thing. But another possibility is that there's size heterogeneity. So the average myocyte is growing, but some myocytes are shrinking and others grow.
So just to summarize briefly for the folks on Zoom, the question was back to the dynamics of growth in the early heart and under pathological circumstances. What role does size heterogeneity play in making those kinds of estimations? Is that a fair summary?
Increase nuclear numbers relative to cardiomyocytes. Well, there's hypertrophy.
Yeah. So first, it's a fair point. These are estimations, and you could only do so much with human tissue, and so I acknowledge that. That said, what Dr. Adler did back in the 70s was to digest the hearts and count cardiomyocyte nuclei. So cell size doesn't play a role. He was counting the nuclei of cardiomyocytes. He showed the nuclei went up in number with increasing mass, and we know that the cells remain mononucleated. And so I still think it has to mean more cardiomyocytes, but we could maybe arm wrestle over this afterwards, because it's evidence. It's not perfect by any means.
Thank you so much.
Welcome. Appreciate that. Thank you.